Hello everyone and welcome to this short video on the biochemistry of sickle cell anemia. Many students have contacted me that they do not understand the biochemical mechanisms that usually result to the formation of sickle cell. So I decided to make. What is sickle cell anemia? This is an inherited disease that usually leads to energy crisis as a result of insufficient oxygen that is supplied to the cells. As we know, when oxygen is supplied to the cells, it is usually used up for catabolism and the breakdown of glucose to release energy. So when insufficient energy is supplied to the cells, it will obviously lead to energy crisis. So at the level of the gene, the sickle cell is a Mendelian inheritance, or it is inherited in the pattern or the Mendelian pattern of inheritance. What does that mean? That means that it follows a regular pattern of inheritance. In other words, it follows the principles that were laid down by Mendel. Okay, that is to say that from the proportion of or from the genotype of the parents, we can easily predict the proportion of the offsprings that will retain the genotype of the parents and those that will not. Okay, it is an autosomal inheritance. That means that it is carried on the non-sex chromosomes. It is carried on the non-sex chromosomes. Okay, which means gender on the non-sex chromosomes. Which means that gender, whether you are male or female, it does not affect the distribution of this inheritance. Whether you are male or female, you have an equal chance of inheriting this disease. It is recessive. This means that we need 2 copies of this allele in order for sickle cell to be expressed. So two copies of the mutated allele is required in order for the sickle cell to express itself. In other words, normal hemoglobin, okay, normal hemoglobin usually represented as hemoglobin A, and the sickle hemoglobin, which is the mutated copy represented as hemoglobin S. So we require 2 copies of this allele, this mutated allele, in order for an individual to express at the phenotypic level the sickle cell anemia, because normal hemoglobin is usually dominant over the sickle hemoglobin. Let's take this example below. Okay, so we take a cross between a heterozygously normal male or a carrier, a normal carrier, and a heterozygously normal female. Okay, so we see that we represent our normal hemoglobin as A because it is hemoglobin A, and we represent the sickle hemoglobin as small s, okay, because it is hemoglobin S, that is a hemoglobin that has the sickle shape. Okay, so we have a cross between this heterozygously normal individual and this heterozygously normal individual or carriers. Mind you, we said that gender does not affect it. So this individual here can be male and this one female, or this one can be male and this other one female, any other, okay, because they are carried on the autosome. So if we do that cross, we will see that 25% of the offsprings will be homozygously normal, okay, then 50% will be carriers, okay, that means they retain the genotype of the parent, and 25% will have sickle cell. So we see 2 individuals that are being crossed, they give rise to a sickle, they have the possibility of giving rise to a child that has sickle cell, okay. So this child is sick because he has 2 copies. Remember, we said an individual requires 2 copies of the mutated allele in order for the individual to express the disease phenotypically, while this individual will not express the disease because of this dominant normal hemoglobin allele that is found here. Okay, so the phenotypic ratio of normal to sick is 3:1. So we see these three individuals. Okay, these 75% of individuals are normal, while just this one is sick. So the ratio is 3:1, and the genotypic ratio of homozygously normal to heterozygously normal to sickle cell is 1:2:1. Okay, so the cross of the parent is usually known as the parent generation, while the offsprings, the first offsprings, are known as the first filial generation. Okay, so what is the mutation, or what usually happens before a normal hemoglobin becomes sickle hemoglobin? Okay, so what happens is a point mutation. A point mutation is a mutation that affects just a single nucleotide. Okay, it is a point mutation that transfers the base adenine to thymine, the base thymine. So this mutation usually transfers adenine to thymine. This results in valine being substituted for glutamic acid. Okay, this results in the amino acid valine being substituted for glutamic acid or glutamate. Okay, I will abbreviate glutamic acid like that for glutamate. Okay, so we see the power of point mutation. Okay, just a single base that has been changed at the level of the gene results to a full different amino acid replacing this one. A full amino acid that is valine replacing glutamic acid in the amino acid sequence. Okay, and if we should remember, when we were doing amino acid, we will see that valine is an amino acid with a hydrophobic side chain, while glutamic acid is an amino acid with an acidic side chain. Okay, so replacing an amino acid of a different group to an amino acid of a different group, that completely changed the function of the entire protein. Okay, leading to a normal hemoglobin or hemoglobin A being transformed to a sickle hemoglobin. Okay, so if we come down here to this sequence diagram, we'll see that this first sequence here is the sequence of normal hemoglobin A, and the one down is the sequence of hemoglobin, sickle hemoglobin. Okay, we see that here we have at position 6 we have glutamic acid. So when that mutation takes place, glutamic acid will be substituted by valine. Okay, that will lead to the amino acid having the sickle shape. Okay, and one thing we should know that mutation takes place in the beta subunit of normal hemoglobin, resulting to sickle hemoglobin, because hemoglobin A, normal hemoglobin A, okay, has 4 subunits. That is 2 alpha subunits and 2 beta subunit. Okay, so this mutation generally takes place in one of the beta subunits. That is why here we see they have put sickle cell in the beta polypeptide. Okay, and here they put beta polypeptide. So this mutation takes place in the beta subunit of hemoglobin A. We have other hemoglobins like hemoglobin F, okay, which is the one that is usually found in the fetus. Okay, instead of these 2 beta subunits in hemoglobin F, we have 2 gamma subunits. Okay, so that is what usually takes place, and it takes place in the beta subunit of hemoglobin A. So when that mutation takes place in that one of the beta subunits of hemoglobin A, it results in the form. So how does the sickle mutation affect the red blood cell? if we look at this red blood cell here, it is a red blood cell that has the normal hemoglobin, which is oval in shape or spherical in shape, and it is flexible. Okay, it is flexible. It's flexible. Okay. Meanwhile, when we look at the sickle red blood cell, this one here, they have a crescent or a sickle shape. They have a sickle shape, and they are non-flexible. They are non-flexible. So how does that affect or lead to sickle cell? Okay, so if we look at this diagram down here, okay, we see that this is the sickle or red blood cell with sickle hemoglobin that has a sickle shape or a crescent shape, while these are red blood cells that have normal hemoglobin. So look at this place here. These are small blood vessels. Because this sickle or red blood cell that contain this sickle hemoglobin, they usually, because they are stiff, they will accumulate on the passages of blood vessels, therefore blocking oxygen from being supplied to cells that are found in this neighborhood here. Okay, cells that are found in this neighborhood. So this will reduce the amount of oxygen that is supplied there, and therefore will reduce the energy that should have been produced through the process of catabolism. Okay, but if it is this normal or red blood cell with normal hemoglobin, they could have easily squeezed themselves through this passage because they are flexible, okay, and can easily squeeze themselves through small blood vessels. But the sickle or red blood cells with a sickle hemoglobin will accumulate because of their stiffness and therefore block oxygen from being supplied to cells found in neighborhoods here. Okay, and this accumulation of sickle hemoglobin here usually also causes pains. Okay, usually also causes pains to the patient. Okay, so evolutionalists, they will say that this sickle hemoglobin should have developed in the tropics. Okay, one thing we note is that the tropical zones. One thing we note is that those with sickle cell are not easily attacked or are somehow resistant to sickle to those with sickle cell anemia are somehow resistant to the malaria parasite. Okay, so the malaria parasite does not easily attack those with sickle cell. And evolutionists, they say that evolution should have occurred that resulted in individuals living in the tropic in order to fight malaria parasite should have undergone this mutation as some sort of a way so that they could resist malaria. Okay, so that is their explanation, that malaria should have evolved in the tropics. This mutation should have developed in the tropics as some sort of a means to fight against malaria. That is why malaria parasite usually finds it difficult to attack those or patients with sickle cell. Now, when you go to the hospital, at times when you have to get married, they will advise you to do this test for sickle cell, and the test that is usually used in the hospitals is electrophoresis. Electrophoresis is a technique that separates molecules based on their charges and the sizes that they have. Those that have a negative charge will migrate towards the positive electrode, okay, known as the anode, while molecules that have the positive charge will migrate towards the the negative electrode known as the cathode. Okay, so this is a gel. They have shown us the direction of migration. Okay, the migration is moving from the positive electrode to the negative electrode. This simply tells us that hemoglobin is positively charged. That is why it is moving from the positive electrode to the negative electrode. So the samples are usually introduced here. The DNA is separated, okay, and then migrated on this cell. So the samples are introduced here. So we see that here we have a homozygosly normal individual has a single band that is found right up here, okay. So this beta A is showing us that this is taking place at the beta subunit of the amino acid. So we have normal, homozygosly normal A. Here we have beta A S. That means that this is a carrier because it has the normal hemoglobin and the sickle hemoglobin. So when someone who is a carrier, when his DNA is run on the electrophoretic chamber, the person will have two bands, okay, one up here corresponding to the level of the normal hemoglobin and one down here corresponding to the band of the sickle hemoglobin. Meanwhile, when the DNA of someone who has the sickle cell disease is run, the person has a single band that is found lower to the position of the normal hemoglobin. Because if we look at the normal hemoglobin here, it is higher, okay. So when your sample is run on the electrophoretic chamber, using the primers for, or was amplified using the primers for normal hemoglobin, when you have these two bands, it means that you are a carrier, okay. And if you have one band that is lower down here, that is to tell you that you have the sickle cell, okay. But if you are heterozygous or you are a carrier and your wife or your partner with whom you want to make a child is also heterozygous or also a carrier, you will be advised not to get married or to give birth, because there's the possibility that the possibility of about 25% or 1/4 that you people will give birth to a child who has sickle cell. Okay, so that is it for sickle cell anemia. Okay, so please, you should subscribe to the channel so that when other contents are being uploaded from this channel, you will be notified, as we'll keep on uploading many other series on scientific topics. Okay, thank you.